everyone, welcome to Aakash. कैसे हैं आप सब लोग? I hope you guys are taking good care of yourselves. This is Lavina Singh, your botany faculty, and today I'm here to discuss the questions from AIATS plus phase 2. We have mock test 3B, right? We have already discussed certain questions from this very test series. So Kirti ma'am told you a lot about many questions and concepts, and I'm here to carry forward with the slot 2, in which we will be taking up the chapters, that is principles of inheritance and variations, and we have a respiration in that. There will be so many chapters that were discussed by Kirti ma'am. Right now I will take up the next slot, in which we will be discussing the chapters such as molecular basis of inheritance. Then we have plant growth and development. It will be a mixture of class 11th and class 12th, because this is for your NEET exam. All right, so very good morning, Kumaran. So I will proceed with this very session, but before that I need to check whether we have the correct question list over here or not. Okay. Alright, so that is absolutely correct. I have already checked it. Now, while we are talking about these discussions, we are thoroughly discussing these test series for you, and many of the students have taken these test series offline or online, right? They actually attempted those tests. Now, this discussion is live for all of you, so that all of you can prepare for your NEET exam very well as it is very important to give all these test series an Aakash touch, right? That is what we are here for, so that you can prepare for your NEET exam absolutely fantastically. All right, so here is the first question today. We have very good morning to all of you. Very good morning, Kiran, Divya. Okay, so question number 99. Okay. Also, there is one important information for all of you. I hope you people are aware of it already. That is, now the number of questions in your section, this botany section, will be 45, right? It has reduced. Entire biology section will be comprising of 90 questions now, right? And all of them are compulsory. There is no leniency for 5 questions that you can simply skip in section B. All the questions are compulsory now, right? So the chapters that I will be taking up, the discussion that I will be doing, will be from anatomy. This question is from anatomy, and let us see what the question says. Question number 99, that is from the test series. Microscopic study of TS, that is the transverse section of a plant part, reveals the presence of following features. Okay, so there are certain characteristic features that are given in this particular question, and we have to tell, after reading these characteristics, that which particular TS do these particular options represent. Okay, we have to see the characteristic features. So the first feature is single layered epiblemma. Second is parenchymatous pericycle. Then we have pith is small, and the presence of 4 xylem and phloem patches. Okay, so the given piece of plant is, so which part it should be? We will begin with shortlisting. Okay, that what should be the correct answer for this. Before coming to the answer, we will simply see that the first point says single layered epiblemma. Do focus on the term epiblemma, right? Epiblemma is the term we use in case of roots, okay? Right, so here we have monocot root, dicot root, monocot stem, dicot stem. So absolutely these two cannot be the answers, because it is speaking of the stem, and we know epiblemma is the term used for the epidermis of roots. Okay, now we have to see whether it is a monocot root or dicot root. Okay, whenever such questions are asked, begin by cancelling out the options, ruling out the options that can absolutely not be true. Okay, so simply go for the root now. Parenchymatous pericycle, okay, then we have the pith is small. This is a characteristic feature for the dicot root, okay. Pith is very small and inconspicuous, okay, and presence of 4 xylem and phloem patches. Now it confirms that this particular these characteristic features are for dicot root, because we know pith is small, In that case, also the presence of 4 xylem and phloem patches. In case of monocot root, the number of xylem and phloem patches are more than 6 in number, right? So there are several. But in this case, the correct answer should be dicot root. But this is not it. We should be knowing the proper structure, all the characteristic features of dicot root, and that I will be discussing with you. Okay, so let us see this beautiful TS of dicot root and understand all the characteristics one by one. Okay, I will be beginning from the periphery and moving towards the center, that is from epiblemma to the pith. Okay, so just make note of everything because these characteristic features are very important. In NEET exam, so many questions are asked around this particular topic. Okay, it can be TS of dicot root, dicot stem, monocot root, monocot stem, or leaves. Okay, any. So these diagrams are very important that are from your NCERTs. Okay, do make a good note of all the characteristic features, all the captions that are given below the diagrams. Okay. Okay, so we have the, okay, as I told you that we will be beginning from the periphery, that is the epiblemma. Okay, so we have epiblemma, the term that we use for root. Okay, then epiblemma, you can see these hair-like structure, right? These are what? These are the root hair that are responsible for the absorption of water and minerals. Okay, so the epiblemma, it comprises of what? There will be the the root hairs will be arising from it. So I can simply mention here we can see the root hairs. Okay, and the characteristic feature for them is they are unicellular. Okay, they are unicellular. Okay, so they are epidermal in origin obviously, and they are unicellular structures. Moving inside, there is the other part, that is the cortex, right? Now cortex forms a major part of this TS. You can clearly see this whole region is that of cortex. Okay, now cortex comprises of parenchymatous cells. Okay, the cells are parenchymatous. Secondly, these cells will be thin-walled. Okay, so this is another characteristic features, thin-walled cells, right? And the other characteristic feature is they are loosely packed. Okay, so the questions can be asked on the basis of these characteristic features also. So, okay, so they are loosely packed. All right, now this is the cortex region. Now what is the innermost layer of the cortex? Okay, do let me know what is the innermost layer of cortex. The question is asked. Okay, so the innermost layer of cortex is nothing but endodermis. Now this endodermis, again characteristics are important for endodermis also. What are the characteristic features for the same? First of all, the layer endodermis is made up of the cells that are barrel shaped. Okay, what do you understand by barrel shape? This type of shape is the barrel shape. Okay, the wooden barrels are there in which grains are stored, in which wine is stored. So such structures that are called as barrel, and these cells you can simply see in this particular representation, they are barrel shaped cells. Okay, so we can say that the first characteristic feature for the cells of endodermis is they are barrel shaped. Next characteristic feature is that the cells are having the specialized thicknings of suberin. Okay, and that is why we can call them as suberized cells. Okay, they can be called as the suberized cells, and this suberin is basically impervious to water. So the endodermal cells that are having these suberin coatings, they will not let the water pass through them. Okay, and they are present in the form of the strips. They form a continuous strip, and that is called as the Casparian strip. That is very important. Casparian strip. Casparian strip is made up of suberin. Okay, it is present where? In the endodermis. All right, clear with this? Very good morning to all of you. Now we have the next layer, एंडोडर्मिस के बाद वी आर स्पीकिंग ऑफ द नेक्स्ट लेयर, दैट इज पेरीसाइकिल. Okay, now pericycle is also very important because questions are asked on the basis of this in terms of secondary growth. Okay, we know that in roots generally the secondary growth is not present, right, especially in case of monocot roots, but when it comes to dicot, secondary growth is there. Okay, and the cells of pericycle, while we are talking, first of all you have to pay attention carefully to this representation, this particular layer inner to endodermis. Okay, this light pink colored layer is endodermis. Inner to endodermis is the presence of pericycle. It can be single to several layered. Okay, so pericycle can be single to several layered. Okay, now what is the function of this particular pericycle? It plays role, basically it helps in the growth of lateral roots and vascular cambium during secondary growth. Okay, so we can simply say that initiation of lateral roots, this is very important. The question has been asked in previous years also. Okay, so please pay attention to this particular point, lateral roots and vascular cambium. Okay, and when will this happen? During the secondary growth. So this will be during secondary growth. Okay, so this is so far the function for pericycle. I hope these are clear with you. Moving on to the next part, that is inner to pericycle, we can see the presence of the vascular bundles. Yes, very, very important. That is the presence of vascular bundles. I am writing it in abbreviation for you. That means we are speaking of xylem and phloem. Now, first of all, we will take up the arrangement of vascular bundles, right? How they are arranged? They are radially arranged. Okay, so vascular bundle is basically radial in arrangement. You can see on one on one radius there is the presence of xylem, either xylem or phloem. They are present in alternate manner. Xylem, phloem, right? So this is called as the radial arrangement. Now, secondly, we can see that these particular vascular bundles, they are 2 to 4 in number. Okay, the characteristic feature that was given in the question. Okay, so that is they are radial, they are 2 to 4 in number generally. Okay, they can be more also, but generally they are 2 to 4 in number. And while we are speaking of their arrangement, इनकी arrangement कैसी होती है? It is exarch. What is exarch arrangement? Basically, when the protoxylem is present towards the outer side, right, and the metaxylem is present towards the inner side. So the pattern will be such. Okay, suppose this is the peripheral part we are speaking of. Okay, let's say that is the endodermal part. Okay, this is the endodermis. So towards endodermis there will be the presence of protoxylem, and towards the pith there will be the presence of metaxylem. Okay, such arrangement is called as exarch arrangement of vascular bundles. So vascular bundles are radial, they are 2 to 4 in number, and their arrangement is exarch. Okay, clear with this? Very good morning, Ishani. Very good morning, Anjali. Now, this arrangement is clear with you, exarch arrangement. Now, vascular bundles are done. Next, we are speaking of the conjunctive tissue. What is this conjunctive tissue? Questions are asked from conjunctive tissue also. Next part, conjunctive tissue is basically the presence of the patches. Okay, can you see? I'll highlight it for you over here. You can see here. There is the presence of this tissue, right? This, is it visible for you? I have highlighted it red in color. Okay, so these are the conjunctive tissues. Okay, can you see their location? They are present between the xylem and phloem, right? They are present between xylem and phloem. So conjunctive tissue. So what are they basically? These are the patches. Okay, they are not in the form of a continuous layer. They are in the form of the patches. Okay, so the patches of parenchymatous cells okay, parenchymatous cells, and where they are present? They are present between xylem and phloem, okay. They also contribute during the secondary growth in the formation of the vascular cambium, okay. Clear with this? So this is the second last part, and the last one, that is the pith, that is the innermost part of this, okay. So the central part that you see over here is the pith, okay. Now pith is basically very small, inconspicuous, maybe not even visible, okay. So that is the pith. So pith यहाँ पर किस तरीके की होगी? That will be small and inconspicuous. Okay, clear with this? So these are the characteristic features while we are speaking of the dicot root, okay. So I hope this is clear with you. So when the such question will be asked, you will be able to answer it. Single layered epiblema, the term that we use for epidermis in case of roots, that is our epiblema. Then we have parenchymatous pericycle. Yes, we saw the pericycle was parenchymatous, 1 to several layered thick, okay. Then we have the pith is small in case of dicot root, and presence of 4 xylem and phloem patches, okay. This is clear with everyone? Okay. Now moving on to the next question, that is the rosette habit of cabbage. Noor Nabi is asking me the chapter number. This is your anatomy in flowering plant. The previous question was from anatomy of flowering plants. Okay. Now here we have question 109, that is the rosette habit. This is the question from your chapter plant growth and development. Okay, that is your chapter number 13. Now the rosette habit of the cabbage can be changed by inducing stem elongation by the application of. There are so many phytohormones in your chapter, there are so many related functions, you have to memorize them all, okay. Especially the functions that has been given in your NCERT, they are of utmost importance, okay. So please do read your NCERTs thoroughly. In this particular question, we are speaking of the rosette habit of cabbage can be changed. That means it is which particular process has been mentioned here, that is the process of bolting, okay. The rosette habit of cabbage can be changed, okay. That means the stem can be elongated, okay. I'll just tell you that what is rosette habit and how it changes, but the process they are speaking of is bolting, okay. When the rosette habit changes, okay, it can be done by the application of which hormone? Can you tell me in the okay, Sagnik is telling in the comment box, gibberellin hormone. Very good. Yes, in beet, cabbage, all the plants that you see in the form of a bunch, cabbage is there, beet is there, right? Lettuce is there. Okay, yes, that is the bolting. Now, you already have answered it in the comment box, that is gibberellin, but that is not all. We need to understand the other functions that are performed by gibberellin, and before that, what is this bolting process, okay. So let's move forward and see. See, there is this cabbage, okay, it is in the rosette habit, okay. It looks like rose, na, that is why we call it a rosette habit, okay. Now this rosette habit, when the this particular plant has to flower, when it has to flower, it cannot stay in such particular texture, okay. The stem has to elongate, okay, and that will look something like this. So this is called as the bolting process, okay. Can you see that? This is the bolting process that has been represented over here. Now bolting, see, first of all, how rosette is formed, you have to understand that, okay. Now if I am talking of a stem, okay, if the presence of nodes is very close to each other, okay, the nodes are present very close to each other, okay, this is one node, I'll use different color for this. This is one node, this is second, this is third, this is fourth, this is fifth. Okay, they are present very close to each other. The internodal length is very less. Okay, that is why the nodes are very close to each other. Now, what we will observe here, the growth of the leaflets will be such, right? Like this. Okay, so it will form a whole bunch, as you can see over here, and this will lead to this rosette habit. Okay, so this will lead to the rosette habit. Now, if I have to change this particular texture, I need to elongate the stem. What I have to do? Simply enhance the length of the internode. Okay, so now the presence of nodes will be far from each other. Okay, so this is one node, this is the other, this is the third one. You can see the internodal length has increased over here. This is the function that is performed by gibberellin. Okay, enhances the length of internode. Okay, now the pattern will be such. Okay. Okay, so this is what you can clearly see in this representation. This is exactly what the gibberellin is doing for the plant. Okay, so when the plant has to flower, it cannot stay in this rosette habit, it has to bolt. Okay, and the bolting is performed by gibberellins. Okay, this is a very important characteristic feature or the function of the phytohormone gibberellin. Okay, so let us see few more functions that are performed by gibberellins, and they are often asked in your exams also. Okay, so gibberellins, first of all, it is responsible. Okay, I switched it. Okay, now first of all, while we are speaking of gibberellins, it elongates, or you can say elongation of stem. This is a function that is performed by gibberellin, as I showed you. It also helps in the elongation of fruit. Okay, for example, apple, to give it a better shape. Okay, so elongation of fruits. यहाँ पे I am mentioning the example also, that is of apple. It has been mentioned in your NCERT as well. Okay, next we see that it hastens the process of, or you can say hastens the ripening process. Hastens ripening process. Okay. First, we will understand hastens the malting process. Okay, not the ripening process. I'm sorry, the malting process. What is the process of malting? Okay, it is a process that is taken up in the brewing industry. Okay, whenever any wheat, any grain is taken. Okay, let's say wheat is taken to form beer. Okay, so what they do, they take the cereal grains, okay, and they form malt of it. Malt kya hota hai? What is malt basically? They take the cereal grains, they are entirely dry, they provided air and moisture. Now what will happen? The seed will begin to germinate. Just begin to germinate. They will not entirely germinate. Okay, they will just induce the process of germination. Okay, so that the sugars and proteins can be broken down in that particular seed. Okay, that was dormant till now. They will just induce the process of germination and then dry those seeds. Okay, now the seeds are not seeds anymore. They have become malt grains. Okay, they are called as malt. Okay, and that is very beneficial for the good quality of alcohol. Okay, now the process of malting, okay, it hastens by the use of gibberellins. Okay, now hastens the malting process. And this malting process in the brewing industry, obviously. Then it also hastens the maturity period. Okay, maturity period in the juvenile conifers. Okay, in juvenile conifers. Okay. And the last but not the least, very important function of gibberellins is they promote seed germination. Why is it very important? Because on the basis of this very function, they are acting antagonistically to abscisic acid. Yes, this term has been used in your NCERT at the last of all the phytohormones that are discussed in your NCERT. There is a paragraph, a line has been given, the antagonistic action of phytohormones. Okay, what is antagonistic action? Basically, when particular hormone is acting in opposite manner, okay, it antagonizes, okay, in the opposite manner to the other hormone. Abscisic acid के लिए gibberellic acid is an antagonistic. So basically, if this particular hormone is promoting seed germination, then what will abscisic acid do? It will suppress the germination of seed. It promotes seed dormancy. Okay, so both are acting opposite to each other. Hence, we say that gibberellic acid is antagonistic to abscisic acid in terms of seed germination, or you can say seed dormancy. Okay, very good, Sagnik. That is the opposite function. Yes, there is another function that has been given of this particular phytohormone in your NCERT, that is sugarcane yield increases, and that is because elongation of stem is being done. All the yield of sugarcane is because of the stem of sugarcane, right? And if the length of sugarcane is increased, of the stem of the sugarcane it has been increased, then obviously the yield will also increase, right? ठीक है, clear with this? Alright, so I hope this much is clear. Moving on to our next question. The question says, which of the given is a hallmark of Watson and Crick's double helix model of DNA? Alright. Himanshu is asking, ma'am, Hindi medium से NEET नहीं हो सकता? अब see, पेपर तो आपका इंग्लिश में आ रहा है ना, और नीट की आप जो तैयारी करते हो वो भी मोस्टली इंग्लिश में करते हो, राइट? सो व्हाई नॉट अटेंड दीज सेशंस इन इंग्लिश आल्सो, सो दैट यू अंडरस्टैंड द कॉन्सेप्ट्स फॉर योर एग्जाम प्रॉपरली. ओके, आई नो हिंदी ज़रूरी होती है, बट यार कभी कभी इट इज़ इम्पॉर्टेंट टू learn the entire session, understand the entire session as well as concepts in English also, right? Okay, now we have the next question, that is, which of the given is a hallmark of Watson and Crick's double helix model of DNA? Now, very important, all the characteristic features of the double helix model of DNA, you should learn them by heart. Very important they are. Here we have been given certain. First is DNA consists of 2 polynucleotide chains. This is true. This is true for the double helix model, but is it the hallmark? Okay, so let us discuss the whole structure first. Okay, let us understand what are the characteristic features of this, the eminent model that had been proposed by Watson and Crick. So while we speak of the DNA helix, this is how it appears like. NCERT diagram, it has been given. Both of them have been taken from NCERT, and you too should. I am taking the references from NCERT so that you can picturize them in your mind and also relate it to your NCERT so that you learn it better. Okay? Very good, Alex. That is absolutely true कि हिंदी मीडियम में नीट तो हो सकता है, but MBBS तो इंग्लिश मीडियम से ही होगा ना, कभी तो पढ़नी पड़ेगी, right? Okay, so let's move to the characteristics of DNA helix. Okay. Now, the first characteristic feature is, while we are talking about the DNA helix, it is made up of 2 polynucleotide chains, right? 2 polynucleotide chains, right? And these polynucleotide chains are arranged in the anti-parallel manner, okay? They are anti-parallel to each other. This is very important. What do we understand by this? Now, in the particular diagram, you can simply see, let us say this is the particular strand that is 5' to 3'. Okay, so the other one will be anti-parallel to it. How is it so? It will be from 5' this side and 3' here. Okay, so 5' to 3', the other will be opposite, 3' to 5'. This is how it is arranged. This is called as the anti-parallel arrangement. Okay. Next we see that we have been asked about the in this question, we have been asked about the hallmark of Watson and Crick's double helix model. Now, I'll tell you about that over here. What do we understand by the hallmark? See, these two, this whole structure is very, it is very important for us. Hamara pura genetic material it is carrying, it has to be stable also, okay? So, it has a backbone, okay? And that backbone is made up of sugars and phosphates. Sugars and phosphates. I'll explain you the structure of nucleotide also in the next question, okay? There is a question in your test paper, many of them have attempted it wrong. I'll tell you that how to correct it and never make a mistake in such kind of question. In that, I will be discussing the structure of nucleotide. But before that, pay attention. This is the diagram from your NCERT. You can simply see over here that this is the one polynucleotide chain, this is the other. This is from 5' to 3', okay? 5' to 3', right? And the other one has to be anti-parallel, so it is from 3' to 5'. Now, you can see there is the presence of this pentose sugar and the phosphate group, okay? You can see phosphate, sugar, phosphate, sugar. This is how this polynucleotide chain is going on, right? So, the backbone of the polynucleotide chain is made up of phosphate and sugar, okay? Sugars and phosphates. Now, these chains are also having the presence of nitrogenous bases, okay? And these are the nitrogenous bases. If the diagram is not clear, if it is too bright for you, simply open your NCERT, okay? You can clearly see the same diagram over there. Adenine, thymine, thymine, adenine. What are these? These are the nitrogenous bases and they are projected inside, right? If this is how the backbone is formed, then the arrangement of the nitrogenous bases is towards inner side, okay? Right? This is how it has been arranged. Okay, so I hope this is clear. The bases are projected inside. Now, these bases are also connected to each other. Okay, they are also paired. That is why we call them as base pair. Okay, so this adenine is paired up with this thymine. So, there is the requirement of bonds and those are the hydrogen bonds. Okay, those are the hydrogen bonds. Now, while we are talking about the base pairing, base pairing कौन करवाएगा? That will be done with the help of hydrogen bonds. Okay, and these bases are projected towards inner side. Now, how is this pairing done? ऐसे नहीं है, you can simply pair anything with anything. Okay, you have to see that the purines are always paired with the pyrimidines, okay? Now, what do we understand by this? You have already studied biomolecules in your 11th standard. We know what are purines and pyrimidines. Purines are adenine and guanine, and pyrimidines are thymine and cytosine, okay? I am talking about DNA here. That is why I am not taking up uracil here, okay? Simply focus on DNA. So, adenine, guanine, thymine, cytosine. So, when the pairing has to be done, it should be between one purine and other pyrimidine, okay? So, I always, when I was in 12th standard, I always used to get confused between this pairing. How it has to be done, whether A pairs up with C or T or G. Hamesha I used to get fumbled. So what happens, I simply learned a mantra that I am telling you. Okay, I'll write it down for you. You also memorize it so that you never make a mistake in this particular question. At the government college. Okay, this mnemonic will help you a lot. At the government college. Okay, so we can simply say A pairs up with T and G pairs up with C. Okay, Now A with T with the help of two base pairs, okay, so two hydrogen bonds. So here will be the presence of two hydrogen bonds, and government college bada hai na, it's huge, so it will require three hydrogen bonds. So this is how you can simply learn at the government college. A pairs up with T, G pairs up with C, and between A and T there will be two hydrogen bonds, and between G and C there will be three hydrogen bonds. Okay, clear with this? Now, this is actually, while we are speaking of this very thing, I just explained, this is the hallmark of double helix model. Okay, so when the question was asked here, the option said, the first option said, DNA consists of two polynucleotide chains. Yes, it is true for the DNA helix, but it is not the hallmark. Second says base pairing between two strands of DNA. This is absolutely true. The base pairing between two strands of DNA, it is being done with the help of the hydrogen bonds, and this is the hallmark of the Watson and Crick's DNA double helix. Purine adenine is equimolar to pyrimidine thymine, okay. I'll just tell you about the Chargaff rule also. And the last one says purine and pyrimidine occur in equal amounts. Now, because this has been mentioned in your option, I'll discuss the Chargaff rule with you. What does it say? I'll pen it down over here, okay? Or I'll simply pen it down for you. What is Chargaff rule? It is very important. The Chargaff rule says that the ratio of adenine and thymine and guanine and cytosine is always constant, and that equals 1, okay? So that means if we are talking about adenine, adenine should be equal, जितने भी हमारे पास adenine के nitrogenous bases हैं, equal amount of thymine is required, okay? Only then they will be able to pair up, right? Okay, thank you so much, Usha. Now we have AT and GC, all right? So A ratio T, or if we are talking about G ratio C, it always has to be 1, okay? So the option said purine and pyrimidine occur in equal amounts, and that is, I told you what is purine. Purine is adenine and guanine, and while we are talking about pyrimidines, that is our thymine and cytosine, okay? So this is how you can keep in mind. They have to be equal in amount, only then they will be able to pair up. Let us say A is 10 in number and T is 5 in number, then remaining A will pair up with what? Okay, so that is why they have to be equal in number. That is what the Chargaff rule says. Okay, the structure of polynucleotide chain is clear with everyone, I hope. Now moving on to the next question. The question says, N-glycosidic linkage, yes, very important again, joins a nitrogenous base with, okay, another nitrogenous base, okay, N-glycosidic linkage. Ab, agar we are talking about N-glycosidic linkage, before that we need to understand the structure of a polynucleotide. So moving further, I'll simply discuss the structure of a polynucleotide with all of you. We'll understand it quickly. Structure of a polynucleotide. Okay. There are three major components for the structure of a polynucleotide. Okay. Yes, we will answer it. We will answer the question, but here the answering part is less important. It is more important to understand the concept first. Okay, Mukesh? So let's see whether you have answered it correctly or not. But before that, we'll understand the structure of polynucleotide. Sorry, a structure of a nucleotide. You people, मुझे ही कंफ्यूज कर देते हो. यहाँ पर आएगा स्ट्रक्चर ऑफ अ न्यूक्लियोटाइड. पॉलीन्यूक्लियोटाइड चेन थोड़ा लंबा कॉन्सेप्ट हो जाएगा. पहले न्यूक्लियोटाइड अंडरस्टैंड कर लेते हैं. स्ट्रक्चर ऑफ अ न्यूक्लियोटाइड. ओके. You have to understand that there are three major components. The first one is a pentose sugar. Pentose sugar. The second one is a nitrogenous base. And the third one is nothing but a phosphate group, right? That is H3PO4. Okay, this is our phosphate. Now, using these components, I'll be drawing up a nucleotide for you. So, here is a pentose sugar, okay? There is the presence of first carbon, second carbon, third carbon, fourth carbon. Where is the fifth one? Fifth is in the form of an extended arm, okay? So, here is our fifth carbon, okay? I'll write it again. Here is the fifth carbon, okay? Now, pentose sugar. Pentose sugar, while we are talking about DNA, we are speaking of the deoxyribose sugar, right? What is why we call it deoxyribose? Because while we are speaking of RNA, I'll make it, Alright, not a problem. I'll draw it again and write it again. Okay, so here is the pentose sugar. Now, while we are talking about the position of the second and third carbon, in the case of RNA, what do we observe? In case of RNA, we see there is the presence of OH at the second carbon also and at the third carbon also, okay? But in case of DNA, there is the presence of only single OH and that will be present at the third carbon, okay? So, one oxygen is gone, that is why we are calling it as deoxy, okay? Ribose sugar it is, but it is deoxy now because one oxygen is gone over here, okay? So, this is the first position, right? And this is our first component, that is the pentose sugar. And this is its fifth carbon over here, okay? This is the first carbon and I'll draw it, but because Sara ka Sara chala gaya, I'll write it down again for you. The first component is the pentose sugar, okay? Now the second one we speak of, that is the nitrogenous base. Now the positioning of this nitrogenous base, because we have, this is the raw structure, we have to attach the next part on this only. So that will be on the first carbon, okay? Okay. A plus T upon G plus C is equal to 1. This is the ratio that they are speaking of, right? The A pair, we are talking about the purines and pyrimidines, okay? While we are saying that the purine is pairing up with pyrimidine, that means let us say a purine, that is A, that is adenine, and it is pairing up with thymine, okay? This is the pyrimidine, right? So, A has to be equal, A should be equal in number to T, because let's say we have 10 adenine, okay, but only 5 thymine. So rest of the adenine will pair up with what? Okay, base pairing has to be done between purines and pyrimidines. That is why their ratio has to be equal. Their ratio should be 1. That means they should be equal in number. That is how the ratio will be 1, right? If they will be 10 upon 5, what will be the ratio? It will be 2, right? We do not want the ratio to be 2. We want it to be 1. So how will it be done? When the both are 10 in number, okay? Only then it will be 1. Understood? That is what we are saying, that the purines and pyrimidines should be having the constant ratio, okay? Now coming back to what we were discussing, the structure of a nucleotide. This is the pentose sugar that we are speaking of. The second component for this will be the nitrogenous base. Okay, now this base can be adenine or guanine or thymine or cytosine, not uracil, because here we are speaking of DNA, okay? So, the position of this very nitrogenous base will be the first carbon of our pentose sugar, okay? So, this is the first carbon, here will be the presence of the nitrogenous base, okay? Now, it will not simply attach just like that. It has to form a bond, okay? And that bond is basically the N-glycosidic bond, okay? I am simply writing glycosidic bond over here. or it is also called as the N-glycosidic linkage. Okay, anything can be asked in the question. N-glycosidic linkage will help the attachment of the nitrogenous base to the first carbon of the pentose sugar. Okay, clear with this? Now, the third component of our nucleotide is a phosphate group. Okay, and which position will it acquire? Can you tell me in the comment box, which position will it acquire? The phosphate group? That will be the last position, that is the fifth carbon over there. Okay, so this is the first carbon, second carbon, third carbon, fourth carbon, and fifth carbon is in the form of this extended arm, and here we'll simply make a bond, right, and here will be the attachment of this phosphate group. Okay, this phosphate group is present over here. Now this bond is named as the phosphoester bond. Okay, you have been given phosphodiester because when there will be the attachment of two phosphates, when this nucleotide will attach to the next one, then there will be the formation of two phosphoester bonds, and then it will become phosphodiester. Okay, but if we have to name this one, we will simply name it as phosphoester bond. Okay, now how the ester bond is formed? Organic chemistry you know better than me, right? That is when water is released. Okay, so over here if we speak of, there is this fifth carbon, right? This fifth carbon, I'll write it over here only so that you understand it better. Okay, so what we see over here, there is the presence of CH2OH. Okay. Okay, I'm writing it this way because over here we have the presence of H3PO4, right? That is the phosphate group. Now, there will be the presence of H3PO4, right? Now, the water will be released from here, H from this, and the water is released, and when the water is released, the ester bond is formed, and because phosphate group is attaching, we call it as the phosphoester bond. What is the bond between phosphate called? This is what I am saying, that is the phosphoester bond. Okay. Is it clear with everyone? Right, now if we are taking up, let's say the phosphate group is not attached. If we only talk about the first two, okay, if we only talk about the first two, in this case it will be called as a nucleoside. Okay, you can see in this particular representation, this is the whole structure of a nucleotide. This is the pentose sugar, this is the nitrogenous base, that is adenine over here, and this is our phosphate group. But this is the nucleotide. If I say this phosphate group is removed, okay, or it hasn't been attached yet, it is called as a nucleoside in that case. Nucleoside, okay? And as soon as nucleoside attaches to a phosphate group, it becomes a nucleotide, okay? So, over here, it will become a nucleotide. You have to keep this thing in mind. Okay, is this clear with everyone? All right, now moving on to our next question. That is, out of 4 major causes of biodiversity loss, which one is mainly responsible for extinction of Steller's sea cow? Okay, so this is the question from biodiversity and conservation. Here the examples are important, the figures and digits, whatever that has been mentioned for you, that is very important. Okay, so also there are 2 graphs in this particular chapter. Please pay attention to them very carefully. Over here, I'll be discussing with you the evil quartet. Okay, what is evil quartet? Basically, the 4 major reasons for the loss of biodiversity, because there are 4 in number, we are calling it as a quartet, and because it is causing harm, it is evil. Okay, so it is the evil quartet. Okay, yes, Sagnik has already given the answer in the comment box. Very good, that is over exploitation, right? But we have to understand the rest of them also. So we will see, first of all, this is the Steller's sea cow. which is extinct obviously, but this is how it looked like. Okay, it was from Russia. And also these locations are important, like dodo was from Mauritius and the stellar sea cow from Russia. You have to keep that in mind. Okay, also remember the locations, okay, from where they got extinct. Okay, so this is the stellar sea cow, and here we have the evil quartet. So what are the four major reasons for the loss of biodiversity? Okay, the first one is habitat loss and fragmentation. Okay, I'll simply explain it to you verbally what it means. Habitat loss and fragmentation. As the name signifies, that the loss of habitat, the loss of home, we are destroying the forest, we are cutting the forest and making, you know, cities over there, we are making malls or roads or highways, so many things, right? So that is the habitat loss, because the inhabitants of those forests obviously will lose their home. That is the loss of habitat. What do we understand by fragmentation? Okay, let us say there was a huge area, okay, the huge forest area, okay. Now some industrialist said ki I want to make a road from here so that the you know, the travel time can be cut down. Okay, so he said I will not disturb anything, I will simply make a road from the middle. Okay, so he said I am simply making a road from here, right? This is the whole forest area over here. Okay, other one came, he said okay, I'll simply build a mall over here, okay, just in this particular area, this much. Okay, the rest is for forest, I'll not do anything, but here I'm making, or simply he is making a huge farmhouse over there, okay, and expanding his whole territory. Okay, now the other one came and said I want to build something over here. I am not consuming the whole area, I am simply taking a small fragment of it. Okay, now what will happen? Basically, this road is crossing this huge forest area, right? Here is one situation and here is the other, okay? Some industrialist has done something, okay? I'm just taking random example. Now, the particular habitat has been divided into fragments now, okay? Let's say this is the fragment, let us say this is A, this is B, this is C, and the leftover is the D, okay? Now, there were some species of animals that were living in the area C, okay? And the species that are living in the area D were dependent on the ones that were living on C, up C. This one, the habitat, the particular inhabitants of the area C have already lost their habitat, okay? So did B and A, they all lost their habitat. But the leftover ones, that is the D, the organisms, the plant species, the animal species that are still living in the area D, they still are facing endangered circumstances, and also they are also losing home. Why? Because they were dependent on C or maybe B or A. Since they lost the particular habitat, they are also not safe anymore. They also cannot sustain there anymore, and ultimately they will also die. Okay, so habitat loss is this particular example. That is, if we speak of this only B section, but fragmentation of the whole area, of the whole forest area, the fragmentation has been done. Okay, so this is what we understand by that. Okay. All right, now moving on to the next factor of this evil quartet, that is overexploitation. Okay, second one is overexploitation. Whenever a particular plant or animal or an organism we are speaking of, if it is overexploited for its goods, for example, stellar sea cow, the fur of this particular animal, okay, fur coats, fur bags, they were very much in demand in France, okay, and because of that, this particular stellar sea cow got extinct because of over exploitation by humans, because of the need and greed of humans. The third one is alien species invasion. Okay. And the fourth one from this quartet is co-extinction. Okay, so the examples for these, first of all, habitat loss and fragmentation. For example, migratory birds. Okay, I'm simply writing single example for each of them. Migratory birds. Okay, or you can say animals. Okay, there are many animals also that do migrate. Let's say 1 year they came to this particular habitat, okay, and they went back. Next time when they came again, they saw that there is no more habitation anymore. There is some big industrialist has made the whole flats or industry over there. Okay, now when it is coming back to this particular area after crossing the whole distance, he is finding, the organism is finding that there is nothing left. Okay, so such organisms are facing major threat. Because of that, migratory animals is the example for the same. Okay, overexploitation is for stellar sea cow, or you can say passenger pigeon, that is also an example. Alien species invasion, here we have the example of Parthenium, that is also commonly called as carrot grass. Okay, so here I am taking the example of Parthenium, that is there in your NCERT. And for co-extinction, see, whenever one organism is dependent on the other, okay, this is very simple logic. A is dependent on B. Okay, A is dependent on B. If B gets extinct, how will A survive, right? So A will also ultimately become extinct. This is called as co-extinction. For example, if we take the example of, let's say, yucca and moth. Okay, so here we have the example of moth and yucca flower. Okay, moth is able to give rise to the young ones because of this yucca, right? And they are interdependent. Okay, yucca gets the benefit of pollination from this particular moth. If yucca becomes extinct, will this moth survive? Right? Where will it lay its eggs? Okay, so moth, this particular moth will also ultimately become extinct, and this is the example for co-extinction. Okay, many pollinators and the pollinating agents, right, that we speak of, and the plants that are getting pollinated, they are dependent on each other. For example, bats and the agave plant. Okay, so if one becomes extinct, the other will automatically become extinct. So that is co-extinction. And this is your stellar sea cow, which was the victim of overexploitation. Alright, so the correct answer for the given question will be your first answer. Okay, fig and wasp, yes, that is also the correct example for the same. Okay, so this is all for today, and in the next session I will be discussing the questions from these very chapters from MT4 test paper. Okay, do attempt it well. All the very best for that, and all the very best for your NEET preparation. Alright, take good care of yourselves everyone. Goodbye.